Good afternoon. Good afternoon. Good afternoon.
Good afternoon, ma'am.
Please put on your audio. Audio, please.
Oh, sorry. Good afternoon,
Yes.
everybody.
Thank you. Thank you. Thanks a lot. Okay, Nilanjana, you can start.
So, I think first the recording would be started by my co-host, Professor
Okay.
Shubhodip.
Recording
Okay.
has been started already. Please
Okay,
continue.
Already started.
okay.
Okay.
Uh,
Thank you.
Dear connections, I welcome you to the day 5, lecture 16 of the refresher course titled Bioscience, Bioengineering and Nanotechnology under the Malaviya Mission Teacher Training Program hosted by Maulana Azad College, Kolkata. As a woman academician from the Maulana family, I deem it as an extreme honor to present before you a power woman of Bengal, our Honorable Dr. Madhumita Manna, the Director of Public Instruction, Education Directorate, Department of Higher Education, Government of West Bengal, Bikash Bhavan. Dr. Madhumita Manna has been a first class holder all through her career. She completed her graduation from the erstwhile Presidency College, Kolkata, and received the coveted Jubilee Postgraduate Merit Prize from the University of Calcutta for her first class first rank in BSc Honours examination. She is the recipient of the prestigious Shikha Ratna Award of the Government of West Bengal in 2020 for her contribution to the domain of education and research. She carried out her research program as a UGC fellow in one of the premier research institute of the country, that is, the Indian Institute of Chemical Biology, Kolkata, and took a PhD degree from Jadavpur University. She developed her research career in the field of molecular taxonomy of Indian Kala Azar and genomics of Leishmania parasites. She joined the West Bengal Education Service in 1998 and served Bethune College and Barashat Government College. She joined the West Bengal Senior Education Service on 2017 as the principal of Bidhannagar College, Kolkata. In 2020, she joined Higher Education Department, Government of West Bengal as Additional Director of Public Instruction in Administration. Presently, she is serving as the Director of Public Instruction of Education Directorate, Higher Education Department, Government of West Bengal, Kolkata. Dr. Manna has completed several research projects from DBT twinning to UGC projects, 3 UGC DAE projects, all from the Government of India, and one WBDST and BT Government of West Bengal. She was a project guide for 2 CSIR SRFs. She has guided 3 MPhil students and 6 PhD students who have received their degrees from Jadavpur University. She is also widely published, and in many of the international and national peer-reviewed journals, she has made her publications. She is also the reviewer of many reputed journals, which include many journals that come under Elsevier and Springer and the like. Dr. Manna has presented her research works at many national and international conferences, symposia, workshops, and has visited Italy, Australia, and Bangladesh on academic errands. She is a life member of All India Cytology and Genetics, Zoological Society Kolkata, and Indian Science News Association. Dr. Manna has been a member of the International Society for Vaccines, USA. She has collaboration with UGC, DAE, SINP, IICB, IPGMER, and so on and so forth for her research works. Dr. Manna's research areas include the genome analysis and molecular taxonomy of Leishmania parasites of Indian Kala Azar, vaccination study with radio attenuated parasites of Kala Azar, nanoparticle-based therapeutics against Kala Azad, trace element biology of the parasites and patients, and the list goes on. Today, she would be talking on technological advances in Leishmania research. And we welcome you, Dr. Manna, and the e-floor is all yours.
Thank you very much for a nice introduction. I'm I'll start giving my thanks to the college authorities, especially Principal Dr. Krishnendu Dotto and the organizers for inviting me to give the talk in this MMTTC refresher course organized by University of Calcutta and hosted by University of Calcutta and organized in Maulana Azad College. As you heard about my little bio note, as Nilanjana told, I work on leishmania and leishmaniasis for a long time, till that date of my research work as a PhD student at Indian Institute of Chemical Biology. And from then on, one field I always carry forward, that is the field of molecular taxonomy and immunology and taxonomy and epidemiology. Later on, as a PhD guide, I developed some other fields like the field of attenuated vaccine or field of phytotherapy. And recently, very few works I have done on nanoparticle-based phytotherapy. Today, I'll talk on my favorite issue, favorite topic, that is the advancement of research by the help of the that biotechnology or bioengineering, what is called. I'll rather say it is biotechnology. And for that, give me some time that I will present you my slides. Okay, thank you.
Dr. Shubhadeep,
Yes, sir.
could you help?
Uh, I think madam's mic is off.
Hmm.
I cannot on it from
Yes.
my end.
Okay.
Madam has to
Okay.
do it by herself.
Hello, madam.
I'm not doing it means
Ah,
I
okay, Shubhadeep,
Ma'am,
please talk to.
ma'am, in
please
your
guide me. I am not very tech savvy. Please guide me.
ma'am, in your meet window,
Hmm.
can you see an icon with a camera at the bottom? An icon with a camera in your meet window, Google Meet window.
I have one share screen. Yeah.
Okay, ma'am. You click on that share screen.
Yeah, that I clicked. Then they are telling that select a tab to share.
No. There are Chrome tab, Windows, and then entire screen. You select
Huh.
that entire screen.
Enter screen I selected.
Then again select a tab to share.
Enter screen I selected. Then I'll tap to share. I am not
Yeah.
share. Yeah, yeah.
Then at the bottom, share button. Okay, madam. Thank you. Your slides are will be now
Yes, yes,
visualized.
yes, yes, yes.
You can see my slides.
No,
Yes.
Yeah, now it is visible.
Ma'am, your screen is visible.
Oh, then
Screen is visible.
what I'll do that
you go to your slides, ma'am.
go to my
Yes,
slides.
ma'am. Once you open your PowerPoint presentation, it will be visible.
Okay,
Uh,
okay.
Yes. Now it's visible. Thank you.
Thank you.
Okay.
Thank you all.
Okay.
So
Okay.
Yes.
can you see me? Means my slides, not me.
Yes, ma'am.
Yes,
Yes,
I
ma'am,
Yes,
can
Okay.
it's
ma'am.
visible.
see.
Yeah.
Lights.
So I'll be talking on Leishmania research, where I am in before going into the topic of today's talk, I will introduce you the world of Leishmania and Leishmaniasis by telling few words or few slides that you can see where the our country, how it has been ravaged by the disease, and how people, thousands of patients lost their lives, that I will show with few slides. So you are looking into a slides where it is written that that in the we know all here the zoology person here all are from zoology.
No,
Madam,
madam, we have a varied backgrounds from chemistry, physics, microbiology, everything.
madam, can I interrupt?
Yeah,
For once, your that Google Meet window you have to minimize.
what does it mean? Oh,
Uh, that means
yes,
on your
yes.
then you open your slides.
Okay,
It is clear.
where I'll do again, it has gone.
Uh,
That is why I prefer always to go for the off-screen mode. Again, all gone. I don't know where.
Ma'am, please click on the PowerPoint symbol
Okay,
at the bottom at the taskbar. Yeah, now the slides are visible.
and I'll minimize that thing. Now it is okay.
Yeah, now it is okay. Yeah.
So I'll click the resume button again or view all slides. Slides show from the beginning. Acha, from the current slides. Okay. Oh, those.
You
ma'am,
may go to
you
resume. Yeah.
Yeah,
yes,
yeah,
now
yeah.
it's
Can
nice.
you see now?
Yes,
Yes,
ma'am,
ma'am.
Yes,
It's
ma'am.
perfectly okay.
So it is, we know the dinosaur became extinct from the face of Earth in that Mesozoic era. No, sorry, in the Mesozoic era, it is dominated by the reptiles, and then mammals came into the picture, and then the extinction of many types of organisms like dinosaur has been, world has seen that thing or have evidence that thing. And at 2004, there is a paper, Poiner and Poiner, they were claiming that maybe dinosaur was extinct for the fact that due to the visceral leishmaniasis, that is the colors are due to that, maybe they became extinct. Now that 4,000 years old Egyptian mummy, by the technical PCR, people discovered that L-Donovani kDNA, I'll come to you about the kDNA, that has been found. And so it is another proof, a very strong proof than the earlier one, that the disease caused many suffering in many from many years back. And then if we come back to Indian subcontinent, here you will see Jessore, Burdwan, Garo Hills, Naugao, Tejpur, Mongoldai, Purnia, Naxalbari. The time period you can notice that 1824 onwards, it has been reported. Surely it was there before that. That the Indian subcontinent was devastated with a particular disease. Nobody that time knew what it was. It is the epidemic report of epidemic in Assam. known as, that time it was known as Assam fever. In medical journalism, Kala-Azar or visual leishmaniasis continues to be referred as the Assam fever. It has many names. That is the Sharkari Bimari, Dum fever, Kala-Azar, Assam fever, visual leishmaniasis. And in Nagaur dispensary, you could see series of that, and many patients are there with their increased spleen and liver. So if we come back to the history, if we say little about the history, the existing medical records telling us the first epidemic of Kala-Azar erupted in 1824 in Jessar district. Now, before that, I have shown you the map. Now in Bangladesh, killing about 45,000 persons in 3 years. And then the Kala-Azar spreaded in Garo Hills, Assam, and from different time point, 1885, 97, 1913, 25, 44, showing a mean inter-epidemic period of 10 to 20 years. Similarly, Bihar has been ravaged by Kala-Azar in 1882 and followed by 1891, 1970, and 1933. Now, that time it is not known what is the causal agent for the disease. The symptoms were so similar with the malaria, and the quinine was not working on the patient. So that time the thought was prevailing that it is a quinine-resistant malaria. And people are dying every day with enlarged spleen, enlarged liver, malaise, anemia, etc. And the British soldier, that time it is the British here, British were ruling here, and in British soldier got that Dum fever. It is named Dum fever because of the fact that those soldiers were working there and going different places of eastern region of the India. They used to come back to the cantonment, Dum cantonment, their resident area, with some fever, unknown fever. As the causal agent and the nature of whether it is parasite or it is virus or bacteria, it is not known. They called it, as it occurred repeatedly in that cantonment, they called it the Dum fever. That soldier, many of the soldiers, they were sent back to England, and that particular soldier, unnamed, he was brought to the Victoria Hospital of Netley, UK, where he died. This British soldier, nameless in medical record or archives, unwittingly gave his life for science. Because Dr. William Leishman that time took up the case for the postmortem, and he has the experience of working in extensively with African sleeping sickness. So that time he found some figures in his slides by the autopsy sample of that particular dead soldier, and he reported that thing to the stalwart protozoologist and Nobel laureate Sir Ronald Ross. He drew the pictures and wrote a letter and sent him. At the same time, this is the letter, I'm sorry. This is a letter of another doctor. who was working in then Madras Medical School, the medical college there. Charles Donovan, he was also working on the same disease, and he also wrote a letter. This is the letter of Leishman, and this is the letter of Donovan. Both wrote almost same time to Ronald Ross about the mysterious structure they found in the blood film. And Ronald Ross, you know all that he got Nobel Prize, and he was working here in Kolkata, in Calcutta, Presidency Hospital, in Calcutta. The new parasite are still a taxonomic orphan. In 1903, Ronald Ross jointly accrediting Leishman and Donovan for the discovery of the parasite and named it as Leishman-Donovan bodies, and taxonomically designated it as a Leishmania donovani, honoring both of them. So what I told you, it is the slide is telling the same thing, that William Donovan from London described the pathogen and published. Charles Donovan from Madras, the other in Madras, made a discovery, and it is the Ronald Ross who gave the name Leishmania donovani. And that gene sustained amastigot and that the Leishmania parasite, it is a protozoan parasite. Protozoa, those who are not from biological background, I'm telling, protozoa is eukaryotes, and it is the kinetoplast. Kinetoplast means that parasite, they have a particular DNA besides nuclear DNA. They have kinetoplast DNA, that is their significance, and that is why they are named as kinetoplastid protozoa. And that parasite has two form of life cycle, two life forms. One is the amastigot, that is the infective stage in the host, in vertebrate host, and other is the non-infective form, that is a promastigot or flagellated form. I'll show you the picture. So, as I told, leishmaniasis is caused by protozoan parasite. Mainly there are three major forms. One is called visceral leishmaniasis, that attacks the systemic organs of our body. Cutaneous leishmaniasis, that occurs in the cutaneous, in the skin, and that occurs in the form of skin lesion. And mucocutaneous leishmaniasis, damaging the mucous membrane of our mouth, nose, the nose, nasal septum. VL is the most neglected tropical disease, and it is fatal. If no treatment is given, it is 100% fatal. Another form of the disease, which is called the Darman sequel of VL, appears, which is very important in terms of epidemiology, in the view of epidemiologists, that is the post-Kala-Azar dermal leishmaniasis. It appears after apparent cure from the VL. It is the lesion, it is in the different form. It is macula, nodules on the skin. And recently, another form has been reported, that is called para-Kala-Azar dermal leishmaniasis or para-KDL. They have both the occurrence, that is the concomitant VL. It is manifestation. It has a manifestation with concomitant VL. Both are in the same patient body. The spleen, livers are affected, and at the same time the skin show the feature of PKDL. So it is called para-KDL. These are the clinical manifestation of different forms. As I told, cutaneous leishmaniasis, it has ulcerated form, disseminated form, diffuse form, atypical form. Mucocutaneous also has mucosal or mucocutaneous form, and visceral. As you can see, the spleen and liver, they are enlarged. If we if we see their life cycle, human being is their accidental host. They have two stages of life cycle. One is in the vertebrate host. Everybody can hear me, could hear me properly?
Yes, ma'am.
Yes,
Okay.
ma'am, you're audible.
Yeah,
Perfect.
Yes,
I'm
ma'am.
audible.
Yes, ma'am.
Okay,
Yes,
thank you. So it has
Yes,
two.
ma'am. Clearly audible.
Okay, thank you very much. So it is the vertebrate host, where I told you just now that amastigote form is prevalent. Amastigote form is found, and it is the most infective form. It attacks the macrophage, that is the organelle, the sentinel of our body. And promastigate form, it occurs in the sandfly. Sandfly is its vector. It is a vector-borne disease. So when sandfly takes blood from infected vertebrate, here the mammalian host, here I have shown that human being. From there it collected the amastigote, and then that amastigote becomes transformed into promastigote, causing the disease. In the meantime, the disease, it attacks the macrophage, and then it starts dividing, rupturing the macrophage, and, distributing the amastigote to attack the fresh macrophage again and again. And then again, that infected mammal is bitten by that sandfly. They take up the amastigote that has become transformed into promastigote in the gut of the sandfly. Then the promastigote multiply and migrate to the proboscis of the sandfly. That means the life cycle rotates. And here in my right side, I'm showing you the beautiful picture of amastigote, where one is red arrow is the nuclear DNA, and the black arrow showing the kDNA. And here the down panel, it is the flagellated form. The interior flagella is present in the promastigote, and it is non-infective form. So if we see the global distribution and occurrence, annually it has been reported that 300,000 VL cases reported annually, and 90% of that VL cases occur India, Brazil, Ethiopia, Kenya, Nepal, Sudan, and South Sudan. In India, then 90% cases occur in state of Bihar. Coming back to Indian scenario, highly affected states in India, whereas I told Bihar, West Bengal, Jharkhand, and Uttar Pradesh. More than 100,000 cases of Kala-Azar occur every year in India, and the state of Bihar alone accounts for more than 10% of the cases. 10 to 20% of the KA patient develop post-Kala-Azar dermal leishmaniasis after the apparent cure from the KA disease in India. Majority of the cases are caused by, historically, I'll come to it in detail, that it is the causal agent is identified, as I told, Leishmania, Donovani, Indian subcontinent, and East Africa. Now regarding the medicine, the chemotherapeutics. The era 1900, that time we have seen the salvarsan discovered by Paul Ehrlich. That medicine got the Nobel Prize. Even a brahmachari developed urea stivamine, nominated for Nobel Prize, but he didn't get it. Alexander Fleming, all you know, discovered penicillin and Nobel Prize in 1945. Domag sulfonamides, she also discovered that thing and got the Nobel Prize. Elvan Waxman, he discovered streptomycin and got the Nobel Prize. So the man whom we will call our hero, but our unsung hero, is Sir Yuven Brahmachari, who developed that urea stivamine, and saved the lives of hundreds of thousands in the British India. And if you see the graph in my left hand side, you will see the drastic decrease in number of death. So we should pay our homage. We means I, me, and all the Leishmaniacs, and all those family who were saved that time. Homage to Sir Upendranath Brahmachari. In each of my talk, I used to keep 2-3 slides to let everybody know what he did. So Dr. Brahmachari was born in 1873, Sardanga village near Purvasali, Bardwan. His father was a physician of East India Railways, and mother is Saurabh Sundari Devi. He completed his early education from Jamalpur, Bihar. He got his BA degree from Hubli Mohsin College. It is a great honor for them that he was a student of their college. He got honors in 2 subjects, mathematics and chemistry. Further, he studied medicine and higher chemistry. In 1984, he got his master's degree from Presidency College. Then he married Nodibala Devi. In MBA examination 1900 of University of Calcutta, Brahmachari excelled in medicine and then in surgery, for which he received the Goodyear and MacLeod awards. He earned his MD and PhD on totally something different subject, studies in hemolysis, both from University of Calcutta. So he has a unique blend of chemistry and medicine, which allowed him to see the problem of Kala-Azar from a different perspective. It is the small room of Brahmachari in the Calcutta Campbell Hospital, where he wrote, I recall with joy, inverted comma, I recall with joy that memorable night in the Calcutta Campbell Hospital, where after a very hard day's work, I found with a smoky, dimly burning lantern that the results of my experiments were up to my expectation, and this little thing would save the lives of millions of fellow men. So the dedication, the knowledge, the sincerity, the love, that made him determined to make the magic bullet. We called it magic bullet, to save the lives of others. What he has done for as I told, he discovered urea stivamine, life-saving treatment for Kala-Azar with a cure rate of over 90% and minimal side effects. Effective treatment of Kala-Azar treatment, reducing the mortality rates and saving the lives of millions. He discovered that post-Kala-Azar dermal leishmaniasis, a new form of cutaneous leishmaniasis, among the Kala-Azar patient. And tropical, he contributed significantly to the understanding and treatment of various tropical diseases, including malaria, leprosy, cerebrospinal meningitis. As I told again and again that he saved lives of millions. He established the first blood bank in Calcutta School of Tropical Medicine 1935. Notable recognition is knighthood. Received knighthood for his outstanding contribution to medicine, honored as Rai Bahadur, and knighthood in 1937. Twice she was nominated for Nobel Prize, and it is our deep sorrow and shame that he didn't get his proper recognition in the form of getting the award of Nobel Prize. So we called him as the unsung hero, but he's a hero. Current scenario, now coming back to the current scenario. In post-independence decades, due to the DDT spraying attempt taken by the center, means to eradicate the malaria. Malaria is also a very big problem of the country. During that DDT spraying, that sandfly, they also die, and Kala-Azar is considered to be eradicated from the eastern part of the country, but that was not the case. In late 70s, the disease came back with full vengeance. The disease broke out in epidemic proportion in the state of Bihar. Adjoining districts like West Bengal, Dadar and Uttar Pradesh were also badly affected. In 1977-78, 4,000 people of India died. In early 90s, 200,000 to 250,000, 250,000 were contracted with the disease. The situation has become worse and further due to the emergence of drug unresponsiveness of the patient towards the first-line drug, sodium ibugluconate. That is, the drug which eradicated the disease once upon a time become ineffective, or patient become unresponsive to the drug, and which is the first-line drug, making the situation obviously very bad. The second-line drugs, amphotericin, pentamidine, panamomycin, where they are very toxic, costly, and less effective. Another problem of the disease was found that as the patient are immunosuppressed condition of the patient made them to contract the HIV. So co-infection with HIV also made the control strategy inadequate. Chemotherapy, as I told, the frontline drug, 60%, now it is not 60%, now it is 70% or like that. Patients are not responding. And the drug, as I told, amphotericin, it has cardiotoxicity, nephrotoxicity, nausea, vomiting, fever. Pentamidine also has a high toxicity and low efficacy. Panamomycin, hepatotoxicity is there, autotoxicity, local pain. And another drug, miltefosine, which is actually a drug for cancer, that was in 2003 or 9, I forgot the time. It has been introduced for administration to the administrating that drug to the drug unresponsive K patient. That was working very nice, but after a few years, that patient also showed the unresponsiveness towards the drug miltefosine. So the current treatment by chemical drugs have several limitations, and cost is also a very important factor. It has frequent relapses and development of resistance. If we talk about the vaccine, I have worked on the vaccine for 5 years. Vaccination is truly a cost-effective way to control the infectious disease. A successful vaccine, as you know, eradicated many diseases like smallpox and polio. It can also be controlled by vaccination, but currently no vaccine is available in the market, and the challenges developing the vaccine is that difficulty in conferring immunity. Kill vaccine have viability concern. Kill vaccine means the parasite will be killed and then injected into the human, not the patient. It is prophylactic, means we use the vaccine beforehand, and that is the prophylaxis. And if we use the vaccine after the disease sets in, then it is the therapeutics. Live attenuated vaccine, I used to work with live attenuated vaccine. It has safety concern, and recombinant or DNase vaccine are still in its initial phases. And adjuvant is to enhance the its efficacy. Some additional chemical is being attached, that is called adjuvant. So that is also not working very well. Now, with the advent of molecular biological technologies and techniques in the field of parasite immunology, molecular taxonomic characterization, genomics and database management system, the field of parasitological study experiences a paradigm shift. And that includes the Leishmania research also. It is the phlebotomine, means the sandfly is known as the phlebotomine insect. That is, they are infected by the female. It should be the female, because female is the blood before the oviposition. So infected female, the disease is transmitted by the bite of that infected female. It is the disease of the poor. In India, the eastern part of the country, as I repeatedly telling you, that devastated with VL. I used to go different endemic areas, so I went in different places of Bihar during my student life, in West Bengal, in student life, in when I am project guide, that time also Jharkhand. Repeatedly I went there. And if we take the West Bengal, 11 districts out of 23 are now afflicted with fever. So somebody used to say that Kalazar is no, it is not now present. So I used to say that data, the 11 district out of 23 is afflicted. Now, as I told, it is some kind of a repetition. Then also I'll tell you the diagnosis and treatment of VL is there, but challenges are there, that the drug resistance, toxicity, lack of vaccine. So global control is at stake. HIV, as I mentioned, it has made the scenario bad. Other species with the disease, association of the other species with the disease also made. What is it? I'll tell you. Leishmania donovani, as I told you, historically known to be the causal agent for the VL or K, but recent observation, not so recent, revealed the association of L. tropica, the causal agent for CL, with VL. That made the control strategy difficult because of the fact that drug for one species may not be effective to the other species. So before going for a drug, for giving a drug, going for a drug regimen, we have to be confirmed about the identification of the parasite. That is called the taxonomical characterization. Proper drug regimen is based on that identification of the parasite. As I told, the drug for one species may not work on the other, and that phylogenetic relationship analysis is also important task for drawing the developing the evolutionary. Now I'll come to you with some technique that is used extensively in the field of Leishmania parasite identification, that is the MLEE, multi-locus enzyme electrophoresis or isozyme technique. And with the spectacular development of purification technique in 60s, that enzymes, proteins, nucleic acid, other complex molecules that had been exploited, the techniques are exploited for taxonomic purpose. Multilocus is MLEE. It is called isozyme profiling, where a population is defined by this profile. It is called the zymodim. Dim means the population, zymo means the enzyme. So what is isozyme? It is done by scoring the mobilities of crude extract of parasite in electrophoretic field of native gel and stain for specific dyes. The gel should be native, means the proteins of the enzyme should not be broken down. It should be in the native state, means in the active state, so that they can be stained with the substrate for that specific enzyme and we will get the band. And it gives a species-specific attribute. The character is species-specific, the profile will be species-specific. Like you see, this will be the zymodim profile. Here I have written, I'm showing you that different enzymes, alert, Azad, nucleoside hydrolase, glucose phosphate isomerase, phosphoglucomutase, mannose phosphate isomerase, ICD6-PGD, MDH. So it is not the single enzyme. We will prepare the crude extract. Crude extract means taking the parasites in a cold condition. We'll rupture the parasite and the soup, and then spin it, and that the soup came out. It will carry all the proteins present there, enzymes, as it is done in the cold condition, and that crude extract, then we apply that in our electrical field. That also kept in cold, and then the migration for some time, that run has been given, the electric current is passed, and after some time we'll stop the electrophoresis and then stain with specific substrate, because it is the enzyme. If the enzyme is there in the lane, it will react with the substrate and it will light up. So in this way, it is not the single enzyme we will take. It is actually we have to take a number of a number of enzymes so that a profile can be made. And that profile is species specific, means I'm showing the profile of L. donovani. So if you get a unknown Leishmania parasite, and if you make the soup, run the gel, and stain the gel, and if it matches with that standard WHO profile, then you can say, okay, this is L. donovani or L. tropica or Amazonensis or Braziliensis or Ethiopica, whatever. Every species has a specific profile, and all the standard Donovani, standard Tropica, standard Ethiopica that are kept in London School of Tropical Medicine. If you want, if somebody interested, if they write, they will give. That profile also they will provide. They publish the small booklet where the procedure is also written. In this way, you can determine the species status of the parasite very beautifully. Here, if you see the NH panel, you will see both both lane has 2 bands each. All lanes has 2 bands, but the migration of 3 and 4 are same, whereas 1 and 2 are same. So they are not the same profile they are showing. Similarly, in PGM, in Azad also, they are showing different mobility. It is the mobility in the electrical field that is being demonstrated here by lighting it with the substrate. Now, Carey Mulisi, he developed the technique called polymerase chain reaction, where a drop of blood, the DNA extracted from a drop of blood, can be magnified, amplified 2 to the power n times. N is the number of cycle the machine will run, the same and again and again and again. So they, after that discovery of the PCR, it has been exploited by the taxonomists, epidemiologists, and population geneticists, using that basic technique in different ways, in their own ways, so that it can be utilized. So introduction of PCR technique has brought a paradigm shift in every field of biological science, and it is mostly exploited technique nowadays. So, and it's especially in the field of species identification and epidemiological surveillance, and the construction of the evolutionary tree. One of the techniques is known as randomly amplified polymorphic DNA. What is it? In the classical PCR, we used 2 primers. Primers means the small DNA fragment which will act as a starting point for the replication. In between the sequence is, if the 2 primer is used, it will sit on the DNA, specific DNA of a particular gene. The primer is developed by knowing the sequence of the specific gene, and they will sit on that by the base pairing. They will sit on that DNA strand, and the DNA strand will act as a what is called, I forgot the name. means it will replicate um, the two primer will sit into end of the that stretch of DNA, and that thing on the basis of the basis present on that old so-called old DNA strand that is used for amplification, the new strand will be replicated starting from the one primer. It will go to the other primer, so that that particular gene sequence will be repeated again and again and again, so that you can do any work, any kind of work with that DNA template. Sorry, that DNA, the older DNA that is used, it is the template. But in case of randomly amplified polymorphic DNA, the name suggests that the primers used here are random. Random means only few information you need or you don't need. One of the few information is that whether the genome, whether it is the GC rich or AT rich, ATGC are the bases. So that much information, if you have, it is okay. If you don't have, it is also okay. So that random primer, people will take that random primer, a particular primer at a time, not two primers, and the extracted DNA from the whole genome DNA that has been isolated, extracted from any living organism, that would be that random primers will be put with that, along with all the DNTPs and the buffer and the magnesium, all that thing. Then if they find matches, that base pair matches, they will sit on the template DNA and they will amplify the region up to a certain length. If the primers sit on the template DNA far apart, they cannot carry on the amplification very good way, or rather it cannot do it. If they sit very close, then also they cannot do. So it is the optimum distance, the primers, same kind. It is not the different kind primers. And they will amplify the genome faithfully in a species-specific manner. That is the beauty of the study. So the classical PCR is modified and used by the researcher in the field to identify, categorize, or taxonomical identification they are doing with the help of many primers, like the isozyme. It is not the single isozyme can construct the profile. It is also not the single primer will amplifies the genome, and then that is the profile of species specific nature. It is not that way. At least 8 to 12 primers should be used with the same genome, and that amplified picture will give you the idea. If you can see the lane number 11 and 12, they are different from lane number 2 to 10. So that is the beauty of the technique. So here, as I told you, L. tropica can cause visceral leishmaniasis or K. That is very unique observation by one scientist from London, David Sachs. In 1995, his group claimed that L. tropica, which is actually related to cutaneous leishmaniasis, and in the eastern part of the country, there is no such co-occurrence of cutaneous form CL or VL, coexistence of these two forms not at all present in the eastern region of the country. It is cutaneous form is present in the western part of the country, in Rajasthan, Himachal Pradesh, like that. But here, he and his group claimed that Indian color can be caused by L. tropica. It has the first such kind of report. Now, my group also reported the same thing. We got one clinical isolate, we named it as T5, and then we have isolated the DNA and we did all the experiment, and we came to the conclusion that yes, it is behaving, giving the same pattern as L. tropica. So that is the finding of our finding of our experiment, and experiment where we will see that all the clinical isolate, 22, 25 in number, all of them are like the L. Donovan standard, but one clinical isolate we got that is behaving like L. tropica. It is another such technique. It is called RFLP. RFLP, it exploits the variation in the homologous DNA sequence known as the polymorphism in order to distinguish individual population or species. So it is the restriction enzyme. I think many of you know what is restriction enzyme. Restriction enzymes are the enzymes present in bacteria which restrict the growth of the virus that tries to infect the bacteria. So that restriction enzyme has the unique property of cutting the DNA strand at a very specific sequence. If the sequence varies, that restriction enzyme will not cut, will not digest the band, so digest the strand. So if any modification occur in the restriction enzyme size, cutting site, that enzyme used in the experiment will not cut it, and then in the electrophoretic run we will see band position in different area. Here you see the 17 and 18. The 2 is the L. Donovan standard from London School of Tropical Medicine, and 18 is the L. tropica standard that we brought from them, or we'll get the gift, we got the gift from other lab. So that amplified ITS region of nuclear DNA, when it is cut with H3 is a restriction enzyme, we got this kind of profile. So 17 is the T7 I am talking about that thing. So T5, though it is isolated from a confirmed Kala-Azar patient, it is showing the closeness to the L. tropica. Another technique is also there, which is called single-stranded conformational polymorphism. It is very simple, sensitive technique, but nowadays it has not been used because it needs that handling the apparatus, that is sequencing gel, is too tedious. And now the science progress so much that the sequencing can be done by giving the sample to any of the vendors, and they will do for you. But that time I did, my student did, and here the principle of SSCP analysis is that based on the single nucleotide polymorphism. That means if any single nucleotide change occur in the sequence, then it will be shown in the native DNA gel. The principal analysis is based on the fact that single-stranded DNA, the strand should be single-strand. We know DNA is double helical structure. and it is, as the Leishmania is eukaryote, it is also the single, it is also double stranded DNA. We have to make the double stranded DNA to single stranded DNA. Then, if there is any change in the nucleotide, ATGC, it will have a defined conformation, and that altered conformation due to single base change in the sequence can cause the single stranded DNA to migrate differently under non-degenerating electrophoretic condition. That is the principle we use for SSCP. So with that SSCP, we are able to isolate separate the drug resistant strain from the drug sensitive strain. That paper, that work has been published. Still another technique which is based on many techniques together, that is called multi-locus sequence typing, or MLST. As I told you that MLEE is the we use there the housekeeping genes, means the genes which are essential for the survival of the organism. They are called the housekeeping. Without that genes, that organism could not survive. Like many enzymes of the Krebs cycle. So if Krebs cycle doesn't go well, or if it stops, the organism will die. So we used to take the housekeeping gene because the evolution of that gene occurs very slowly. So if you want to see any change in the species level, we will take the sequence which is more or less conserved. Any deviation from the sequence will tell us that here some variation occurs, and that may result in different strain, different species, like that. So here, like that MLEE, in case of MLEE, we have used the different enzyme, means protein. The beta technology came up when people start using not the protein but the DNA. So here also again, the DNA is isolated from housekeeping genes, and then the sequence is amplified, PCR amplification. After PCR amplification, it is sequenced, and then alignment. They are doing the alignment, and all through machines. So the workflow of the MLST involves the data collection, that how many sequences we are getting, data analysis, and multi-locus sequence analysis with what is called ST type. So in doing so, it is the basic plan I am saying, the sequence, the alignment, the sequence type, assignment, that is the scheme. And in doing so, we see that alert is the enzyme, already I told, alanine aminotransferase, as I told in the gene of alert. There in MLE, you have seen the enzyme alert, the protein. Here the gene of alert, that has been sequence typing and by the MLST software. And then we have developed the tree, and that sequence that shown, that ST3 is exclusively present in T5, that is the L. tropica clinical isolate of Indian Kala-Azar patient. So here we can illustrate the interspecific genetic diversity and unique sequence typing. We can show it, and in particular display a high nucleotide diversity among the coding region of the high scoping genes throughout the genomic sequence. L. tropica isolate had unique ST3 for alert, and for enol is another enzyme local. same STC was shared by two donovan is T3 and T7 isolates. And here again, the Indian Kala-Jar isolates can be differentiated from the para-KDL isolate by sequencing typing. I forgot to mention what is the role of PKDL and para-KDL. I'm sorry for that. PKDL and para-KDL they act as a reservoir for the parasite during the interepidemic period. Here, many other reservoir are discovered in different areas, endemic areas of the leishmaniasis. But here in India, like goat, like jadbil, like cat, dog, many reservoir are there. But in India, we have not identified or noticed any reservoir, and it is said that PKDL and para-KDL they act as the parasite lies beneath the skin, and, uh, and that is the manifestation of the disease. So parasite remains silent there. They don't cause any virulence. If they cause virulence, the patient will die. Parasite also know whom to they keep alive and whom to. They will never make anybody die because they need the host, need the reservoir, and they don't do any harm to the reservoir. So that reservoir, PKDL, they are all anthropomorphic reservoir. So anthropomorphic means human being. So para-KDL and PKDL are very important stages of disease, that particular disease, that should be studied very carefully to control that disease. But that control strategy is nowadays depends on strategy depend on the control of on the biology and the immunology and the disease progression of PKDL, para-KDL. They are very, very, very important. Fortunately, we got few para-KDL patients during our stay at Birbhum in Jharkhand. From Jharkhand, we collected that para-KDL. PKDL also we have. Drug-resistant parasite, we have also one miltepocine-resistant parasite we got. So here is the importance of para-KDL, and we have done a lot of work on para-KDL. Now I'll come to the genome. Genome means Leishmania genome and the whole genome sequencing. Here, as I told earlier, that that Leishmania genome is unique. Better to say trypanosomatid genome is unique for the presence of two DNA moiety. One is the kinetoplast DNA or kDNA, other is a nuclear DNA. It is a mitochondrial DNA structure, Leishmania and other trypanosomatid. It has maxi circle of 20 kb around size, and it encodes the rRNA genes and the structural proteins for the mitochondrial respiration. Mini circles are many in number. It is 0.8 kb. Encode guide RNA, gRNA for editing the maxi circle encoded mitochondrial genes. And nuclear genes is a parasite Leishmania is a diploid genome with 34 or 36 chromosome. Genome size is 0.3 to 3.5 MB. It is rich in genes. 8,000 to 10,000 protein genes are there, organized in a polycystonic transcription unit, meaning multiple genes are transcribed together as a single unit. Here, it is a next generation sequencer setup where we can sequence whole genome of parasite. So our strategy was to take the SSG means SAG means sodium stevogluconate, SSG sensitive VL, SSG resistant VL, SSG sensitive para-KDL, 3 and 1 mil resistant. Above 3 or with 3 each, and we got only 1 mil. Mil means miltepocine resistant. That is, we want to sequence all the whole genome of all the parasite we have that time in our hand. So whole genome analysis, we had unemployed mutation profile, copy number variation, I'm not going detail into it. The plan is that that SSGR sequence, and there will be the genome sequence will be done, and what information we will get, that we'll see. So we'll get the 61 mutated genes across the whole strain studied. Mutated in the pathway analysis, it is also a software-based database analysis. As I told, that different techniques arrived, and many database management system arrived. We know GenBank accession, we know NCBI. So using that software, we can say that the genes we have seen that mutated, what are they for? What for? They are, sorry, that the functionality of the gene, the activities of the genes we can understand. So some are metabolic pathways involved in metabolic pathways, some are the biosynthesis of the amino acids, some are biosynthesis of secondary metabolites, some are purine metabolism, like that. So it is also another very good set of information we are getting by. Now, when we the SOMI value means trisomy or tetrasomy or like that. So we have seen, as I told, each strand contain on an average 30 disomic chromosome, that Lishwania donovani. SOMI variation was found in chromosome number 8, 23, 26, and 36. Paracardial S strand means sensitive paracardial. 24 gene mutation are unique, so we can work on it. We will be able to understand the mechanism of controlling them. Tetrasomy means four sets, that one chromosome in 4 in number. That is in VL SSGR means resistant strain. SSC resistance is chromosome aneuploidy non-synchronous genetic variation in the coding sequence. I'm not going detail into it. And tetrasomy of chromosome 23 and 1 pentasomy. These are all the findings we are correlating with our clinical isolates and its biology. These are the softwares: NCBI BLAST, Makeup, MLST software, DNS, V6 software, rplot, KEGG, PyMol, 3D structure, secondary structure, metabolic pathway study, heat map construction, genetic diversity indices, all are now available in the fingertips. That is the beauty of development of the science and the technologies. What is the time? May I continue?
Yes, ma'am, of course, if you wish to. After that, we can take the questions.
Okay. Here now, that is the identification part I have covered. Means the work I used to do I have done in my lab, and nowadays the characterization part I'm not doing because of my busy schedule as administrative work. Now, green technology and nanotechnology, it is a sustainable alternative. And what is the green? It is the control. The first part I told, it is the identification of the parasite. If we identify the parasite, next we'll go first we identify, then we'll characterize, then we'll see what is its condition, what are the unique sequence or unique genes are present, then we'll target it for the drug, so that the drugs used for the killing them should not kill the host at the same time. So it is the green nanotechnology. Before coming to the nanotechnology, I have done some other means phytotherapeutics. That is the medicinal plant as an anti-leishmanial agent. Before doing that, entering into that nanotechnology field, that I have not done much. So here we have done some work on phytotherapeutics with the medicinal plant and some later on some mangroves. These are the plants. Saraka Ashoka, it is the Ashok full. Ficus racemosa is the fig, Tomur. Cinnamomum tamala is bay leaf, Tejpata. And Coccinia grandis is Talakuch. So these are our indigenous available here and there. And already they have reported, there are reports of anti-malarial, antioxidant activity, anti-cancer, anti-inflammatory, antimicrobial, hepatoprotective, anti-diabetic. Like these are all known facts. What we did, we take the twig or flower or leaves or bark and then dry them and then grind them and then prepare the ethanol extract or methanol extract. It may be ethanolic leaf extract of Coccinia grandis. So here the key point of the line is that in our experiment in mice model, biopsy mice model, what we used to do, we used to keep the control group, giving nothing to them, not the parasite, not the treatment. Another is the experimental group, 3 or 4 or whatever. And then the infected group, that means they got that group of 6, 7 or 10 mice in a group. They will get only the infection, Leishmania infection through the heart, like that. It is very difficult to do, but I work in a lab in ISCB, so that is we some of the people are there. They are very expert in handling this thing. So in this way, the infection is set up there, and then we use that experimental groups. We use the extract. We standardized that thing. So Coccinia grandis, LE means leaf extract of Coccinia grandis, ethanolic leaf extract. And in killing the parasite, the nitric oxide and the ROS species, they are increased significantly because they help to kill the parasite. And so that N1 ROS level of these treated groups are increased significantly in respect to the infected control group. It can fragmentize the DNA, depolarize the membrane, mitochondrial membrane potential, which ultimately leads to the apoptosis. And these leaf extract, they act both on the drug sensitive and drug resistant strain. Like we have done similar kind of experiment with Ficus racemose leaf extract of ethanolic extract, L for leaf, E for ethanol, and Cinnamomum tamala. That also we have done. We have done the same kind of experiment, taking different extract part, different plant parts. In this way, we have done many work. Saraka Ashoka, when I was at Bethune College, I have taken the flower of Saraka plant there and started doing that thing. So here also the same thing we have observed. Here also we have also observed that TH cytokine. TH cytokine is the cytokine Cytokine is smaller group of chemicals that is released in the body, in our body. When we are parasitized, our Th2 level or another kind of cytokines that is increased. When it is we are recovering the Th1, and that I'm talking about the mice, the Th1 cytokine helps in parasite killing, whereas the Th2 cytokine helps in the disease regulation. And it is seen that EBSA extract treatment was able to enhance the Th1 level in treated groups and decrease the Th2 level. So that means it has a leishmaniacidal activity. So phytotherapy offers a promising alternative to conventional treatment for leishmaniasis. That it utilizes the medicinal plant native to specific region. So it depends on that our we take help from that traditional knowledge. Our study plants exhibit potent activity against the Leishmania parasite, both in drug sensitive and resistant. Herbal formulation is always better than the chemical formulation because it has very reduced side effect and cost effective. We have also used better I stop nano. I think I cannot.
Yes, ma'am, I think.
So I am. I'll stop here. Huh?
Okay, ma'am. Okay.
If it is requested, if time permits, some other day I may talk, or for this time I am not taking that nano part.
Wonderful. Or, ma'am, it can even come in form of questions. We can take them if, these parts come as a part of your question.
Means they will question me. Let them do.
Yes, ma'am, they have already started questioning.
Eh?
And yes, thank you, ma'am, for that loaded presentation on this Saturday afternoon of December. It
Yeah.
was packed with information which are of, social, medical, environmental relevance. It also was packed with knowledge, ma'am, as you demonstrated your intellectual commitment to make a researcher like Dr. Brahmachari visible in the history of Bengal science. And thank you for those captivating slides. The questions have started coming, and if you permit, I would start reading them down. The very first question had hit the presentation, which is directly from Sundarban, and she is Manasi ma'am. She asks you, as she's from Sundarban Hazi Desarat College, ma'am, why would a person suffering from protein energy malnutrition be more susceptible to Kalazar? As you mentioned, that it is the disease of the poor. This is the first question.
Okay. Now that diet, that protein diet, that boosts our immune system, boost our mineral system stronger. It and if we are deficient of proteins, that malnutrition will not get enough zinc, enough magnesium, enough many things, and that you cannot means that immune system will be weak. That is why that malnourished people, they catch up the disease so easily. And the
Thank
disease management also, as they are poor. Another very interesting fact, the question is very nice. Another important factor is that for the treatment of the disease, the patients have to be admitted in the hospital for 28 days to get the full course of like, say for example, sodium stevogluconate or amphotericin B, or miltefosine. So for a poor person, it is not easy to remain in the hospital condition for that many days. So after apparent cure, they left the hospital, and they go back as a human reservoir. So that, and the social status also means they are not aware because they are not that way educated. They used to go to the forest to cut the woods, and in the mud house, the wall, that crevices of their mud house wall, that is the egg laying area for the sandflies. So there are many socio-economic condition also. That is why poor are very prone to the disease.
Thank you, ma'am. She again
Okay,
asks
ma'am.
you,
Thank you.
she also asks you, is there any modern diagnostic kit by which early detection of leishmaniasis is possible?
Yeah, it is there. Rapid test they call RK39, like that. They use in a field, and in the field also there is a PCR technology also used in the field, not in our country, um, to do the early diagnosis. So there are there. These are there.
So I think you have been sufficiently responded by ma'am. And now we move on
Ma'am,
to Mr. OP Singh. Yes, Manasi?
ma'am, I want to ask you, is this available in our country, particularly in the primary health center like that?
RK39, I think, available. Actually, the disease is the neglected tropical disease. Huh? It is not bringing many funds. It is only the WHO and the government setup that is taking care of the disease. In Jharkhand, I have visited a hospital there. They have developed a very nice system. They used to send their what is called oh, that ladies, they are working, Asha. The Asha people, they used to go to the villages. For that, they are given the cycle to the villages. They used to go there and feed the patient with the medicine. Otherwise, they are very reluctant taking the medicine. In this way, Jharkhand, they have eliminated that disease to a great extent. To a great extent.
Okay, thank you, madam.
Thank you. The second question is from Mr. OP Singh, and he asks, how does Leishmania donovani alter host enzyme activity during parasitism in animal models?
It is a big topic. Please give my number to him. Means here, after a long talk, I cannot discuss the mechanism. So it is the development of the that they close it. The parasite down regulate the TH1 synthesis and upregulate that thing. So this is very complicated way that needs another class to. So that when that parasite overpower us, we are parasitized. When the macrophages, the release of the cytokines, that NO and ROS, they are coming into the play. If they overcome, then uh, the patient survives. So it is very complicated pathway there. That is for the timing. That is my answer.
So,
Thank
sorry,
you.
madam, sorry to interrupt you.
Yeah.
So, I am Dr. OP Singh from Banaras Hindu University.
Oh, nice
So,
meeting you.
uh, it was a very great talk, man. My question is actually regarding something different. So, the thing is that the visceral leishmaniasis is considered as a anthropogenetic in nature.
Mhm.
The disease nature is, but now with some of the research publication, there is a question raised that the parasite can also reside in the some of the animals.
Yeah,
The,
from Nepal it is there. That
yeah,
is the goat is infected, but here
yeah,
only Kilikendrick once found when she was traveling Bihar, Daden Bihar.
yeah,
She got one jackal lying on the dead on the road, and she took the sample from there, and she told that jackal is the host. But that is a very scanty report in India.
Exactly.
But some reports are there that some of the vertebrate hosts, some of reservoir are here. In Nepal, it is the goat, as we know, as you know, as I know.
Yeah, so my question is that, ma'am, because if there is a animal reservoir possibility in Indian subcontinent, so this might be pose a real challenge in the VL elimination. So the thing is that if I isolate the parasite from any of the organism, can we uh, uh, can we have some any molecular tools by which we can say this parasite have the ability to survive in the animal or not, so that we can provide some of the molecular information to the our policy makers, that yes, this is the some molecular techniques by which we can say that the parasite which are circulating in the Indian subcontinent, they have the ability to survive in the animal.
No, that's kind of report I have not come across, very precisely speaking. If you have come across any such report.
That's the question, because I am also the searching of the some of the method by which we can provide the information.
No, that whole genome sequence of the parasites giving us the mutated genes, as I told, that paracadial. So they are the mutated genes. If they are related to, I have not done that the next part of the work because of my extreme business. That if that thing we could do, then there is a chance we can predict that when that parasite changes itself from veal to picadile, that is the very sensitive place which we can put a checkpoint. That still I have not done, or not anybody have done so far as far my knowledge goes.
So, Dr. Sinha, what we can right now do is that, as ma'am has already requested us to give her mail ID to you, so you can collaborate with her, and there is a lot of space and scope for you to now, think together and dream together.
Sure,
And
ma'am.
with
আর
that,
কটা প্রশ্ন আছে, আমি একটু তো জল খেতে পারি নাকি, একটু জল
ma'am,
খাবো?
please have water. You need voice rest.
Yeah. Okay, I'm coming within a
Sure, sure.
minute or one. Sorry, thank you.
Thank you, ma'am. And so that shows how involved she was as a speaker today. And we have another two questions from the chat box. First is Dr. Rakhi Chakraborty's question. That is, are there any reports of anti-leishmanial effects of plant secondary metabolites?
Yeah, that is there. Lots of secondary metabolites are used nowadays, trying everybody. And according to HO, without people better do research on plant products to cure the disease than to do the immunology or molecular biology or whatever science. So all are trying, and secondary metabolites are there. Many reports, you go through the your literature, you will get a lot of it.
Thank you, ma'am. Thank you so much.
Okay, then.
I think with that, yeah, we need to wind up. We can take, I think, one last question, if ma'am permits. That is from our very Maulana family. Dr. Rajarshi Ghosh has asked, what biomarkers of Leishmania infection can AI help identify from blood tests or molecular data? Example, PCR results. And with that, we would be ending the session.
কি বলে রাজস্বী, তুই একটু ভালো করে বুঝিয়ে বলো তো কি বলতে চাইছিস? এরকম একটা বিকট প্রশ্ন করে দিলে, হোয়াট ইজ ইট?
হ্যাঁ, গুড আফটারনুন মধুনাদি। যেটা আমার বলার
হুম।
ইচ্ছা ছিল যে এখন তো এআই এর যুগ, এআই এর
হ্যাঁ।
এরাতে নতুন কোনো ইনফরমেশনস বা ও কোনো ক্লু দিতে পারে কিনা যে প্রপার আর কি
ইংরেজিতে বলো, ইংলিশে বলো, talk in English. Sorry that I have started talking in Bengali. Sorry for that.
Okay. So that's why I too. Actually, the applications of AI has been immersed in a wide scale. So in that era of AI, so does AI possess any tool to diagnose the, I mean, the leishmania
So
or
far,
the
I understand. So far, my knowledge goes, that kind of report we have not seen anywhere. Maybe it will come up, or maybe I have not seen. But till date, I have not been reported by my students also that, Madam, read that publication. That is the case. Okay.
Wonderful.
Okay.
So that had
Thank
been
you.
very interactive session, and we would like to thank the organizing body led by the coordinator, Dr. Deepak Som, and the assistant coordinators, Dr. Shubhodip Samanta and Dr. Rajanta Mukherjee. Of course, our honorable DPI ma'am, who has shared her literally valuable time with us, the resource person of the day. And we also thank our principal, Dr. Krishnendu Datto, the IQAC coordinator, Dr. Shampa Datta Gupta, and to each and every participant for their intellectual, active participation today. That was a wonderful session, and thank you so much for being here.
Thank you.
Bye, ma'am.
Bye,
Take
bye,
care.
bye, bye, bye. Thank you, everybody.
Thank you, madam.